Hello, everyone. My name is Joe Magliozzi, and I'm a postdoc in Goode Laboratory at Brandeis University. And I wanted to give the organizers some thanks for giving me the opportunity to talk about my journey in science, as well as what I do as a scientist now. And so today I'm going to be walking you through how I became the scientist that I am today, and a little bit about the type of work that I'm interested in and how we conduct that in the laboratory. So this all began for me actually just a few miles up the road in Burlington, Massachusetts. That's where I'm originally from, and this is the high school that I attended. And my journey through high school was really focused on trying to figure out what I wanted to do. Probably something similar to what some of you are facing now is, what path would you like to take in establishing some form of a career? And that led me towards actually the a career in business. My high school was very oriented towards elective courses in accounting, marketing, business management, amongst others. And that is where I thought I was going to pursue a career. But as I attended different college tours and sat in on sessions about careers in accounting, amongst others, I didn't see myself actually doing that. And where I really started to find my way in comfort in what I could do in the future, um, I was really motivated by a biology teacher I had for both regular biology as well as AP biology. This teacher, Mr. Wood, who I actually wrote my college essay about. And he inspired me to pursue science in that it's not as daunting as it sounds. And if you're really invested in the topic, it will be a very fruitful endeavor. So I got into college. I ended up going to Virginia Tech for my undergraduate. This is located in Blacksburg, Virginia, for those who don't know. And I majored in biology. And a lot of the people I was going there with were pre-med, pre-vet. But I didn't want to do that. I was much more interested in the scientific method and understanding how to use science to answer interesting questions, which led me to joining the research lab of Dr. Takashi Fukao. But don't get it twisted. I applied to a lot of different laboratories and faced a number of rejections and a number of professors that didn't get back to me about joining labs. So this was a difficult egg to crack initially, but Dr. Fukao was a new assistant professor, and he generously offered me a position in his lab where we studied the role of gene expression when you submerge, in this case, soybean as well as other crops to flooding conditions, which is very important because all across the world, flooding can reduce crop yields and thus lose different farms, upwards of millions of dollars per year. So understanding how these genes affect the crops can help us create versions of plants that may be more tolerant to flood conditions. So what we learned is through my research is that plants adapt to stress and change their gene expression during water submergence. And this work with plants made me extremely interested in pursuing plant biology further. So I then ended up applying to graduate programs, research-based programs that had a heavy emphasis in plants. And where I ended up going was Dartmouth up in Hanover, New Hampshire. So not too far from here, a couple-hour drive. And so when you go to graduate school, oftentimes you go through these what are called rotation periods where you join a lab for 10 weeks or so, and then you do that two or three more times. And then at the end, you try to come to an agreement with the lab to join full time as a PhD candidate. I started in a plant lab, but through my rotations, I tried different flavors of science until I ended up in the laboratory of Dr. James Mosley. And he actually does something very different from what I was doing as an undergrad. He studies how, using a model organism called fission yeast, how factors that regulate the growth of the cell actually influence the division of the cell, which are often viewed as diametrically opposed processes. But what we figured out is that if you don't coordinate growth with your division, cells can undergo aneuploidy, which is where you get different chromosomes or DNA segregating where it's not supposed to go. And this is a hallmark of forming cancer cells. So this work, though in a model organism, was rather fundamental to things we care about in the healthcare world. And my love for science made me want to pursue it one step further after my PhD and do what's called a postdoctoral fellowship. So this is research that happens after your PhD, and it's to establish a new independent path that you would take with you to start your own laboratory if you choose to go and work in a university or a research institution. In this position, I'm the postdoc in Bruce Goode's laboratory, who's at Brandeis. And what we study is how cells organize their interior. So here is your classic textbook example of a cell, and you have your different organelles, like the nucleus and the mitochondria, outlined in this easy to see schematic. But that is not actually what a cell really looks like on the inside. And what does the inside of the cell actually look like? And our focus inside of the cell is what's called the cytoskeleton, or the skeleton of the cell. And what I'm showing you in this image are these different protein polymer networks. So they're these proteins of different natures that actually come together and form filamentous structures. And they form these beautiful networks that not just look pretty, but they actually provide function by delivering things to different parts of the cell, maintaining structural integrity, and allowing cells to grow and divide properly. And my research focuses on one particular branch of this cytoskeleton called actin. And actin mutations are linked to a number of diseases, and actin is one of the most highly expressed proteins in all of your cells. So for these reasons, it's a very active area of research in trying to understand the roles of actin biology. And to do this work, I use another model organism called budding yeast. And the budding yeast is comprised of a larger mother compartment that grows by forming what's called a bud. And this budding process creates a smaller cell that will grow to a certain size. And then at some point, the cell will go through division and will pinch off and will repeat the process again over and over. This system is great because it's easy and inexpensive to grow. The genes that are expressed in budding yeast are remarkably conserved in humans, and we can make genetic mutations far faster and more readily than we can for human cells. And the actin in budding yeast is primarily divided into two networks. These dot-like structures are called patches, which I won't talk about for this talk. And then there are cables, which are these long filamentous networks that extend from that bud compartment down to the back of the mother cell. And these serve as tracks for delivering growth factors to allow the cell to grow properly. And if you don't have either of these structures in your yeast, the cell is not viable and it will die. And yeast actin shares a remarkable 90% identity to human actin. And the proteins, again, required to make yeast actin are highly conserved in humans. So for all of these reasons, we can take a very simple system and answer large, spanning questions across the eukaryotic cell kingdom. And I'm interested in how these actin networks are made in cells. So what I have here is a movie playing how we think this works in yeast. Those long networks that I showed called cables are generated by two separate proteins called BNI1 and BNR1, and these are classified as formins. And all you need to know is that these formins are required to generate these long networks. And like I showed you before, but here I'm actually showing a movie of it, these cables are there to grow and then latch on these growth factors that get trafficked up into the growing bud. And here's a better example of how formins work at the molecule level. So the formin will come, bind to actin protein, and then incorporate it onto its growing end of the filament or cable. And this occurs processively, so it will stay and grow on the end. But how do we study protein function in the cell? And this is the essence of how both the Goode Lab and I approach answering these questions. We take what's called a two-pronged approach. So we start either direction, but in this case, we'll start top down. And what top down means is that you can take a cell and you can make a mutation and then see what that mutation does to your process or structure of interest. So here we have our actin in a wild type cell. So this is a normal cell, and you can see you have the dot-like patches, and you have the bright filament, the bright long cables. And then you have your mutant, which you can see by looking at the image has much thinner and less organized actin cable networks. So this can give us this can allow us to give the conclusion that knocking out or getting rid of certain genes have fundamental consequences on the actin in the cell. But where we really go deeper is from the bottom up approach, where in this case, we're coming at it from the opposite angle, where we're taking the purified molecules and we're doing experiments to see what they do in isolation, and then combine that with our top down approach to cohesively generate a model that how we think this stuff works in the cell. So what we can do is we can label our actin in the test tube, and then we can add in labeled regulatory proteins. And in this case, we're seeing how an actin binding protein can bind actin, coat the filament, and actually remodel its organization. And in a different assay, we can go even more granular and we can watch single filaments being generated by a single molecule. So we have our uh, pink dot, that's a single molecule of a particular actin binding protein. And you can see it's actually assembling the actin from one site. So we can go all the way to the level of a single molecule up to looking at an entire cell. And these approaches allow us to understand how molecules organize structures in our cell to allow it to grow and divide properly. So, in summation, science as a career is difficult, but it is ultimately very fulfilling. I have found that through publishing my findings in different journals, attending conferences, building a scientific community has been a very rewarding experience. And as you can see from the word cloud I generated, these are from titles of all of my different publications. And there's a lot of different words in here that are unrelated to each other. And it was nice to look at this and think about all the different projects I've worked on and contributed to, and hopefully will continue to do in the future. And I will leave you with the last final quote, which is to be a good scientist, you don't have to be perfect, you just have to be good. And that is from my high school teacher that I mentioned in the beginning of the talk. And he used to say this a lot during class when we were going over rather difficult concepts, because I think sometimes science can get put on this pedestal of being so difficult. And I would say that persistence and interest can far outweigh, you know, the ability to understand something on the first try. And it was that sort of advice that led me to fundamentally changing what I was going to do for a living. And I'm very happy with the choice that I made. And so thank you for your time.